Well, welcome back, and I hope you had a great stretch and are ready for the next set of sessions that are sponsored by the Turkey Farmers of Canada. Our first session is broiler related, and I'm happy to introduce Mary Lou Gaucher from the University of Montreal. Her title is Evaluation of Immunogenicity of Putative Antigens Identified in Necrotic Enteritis Causing Clostridium perfringens using reverse vaccinology. So with that, take it away, Mary Lou.
Thank you, Val. I'll just share my presentation. So do you see my presentation?
Yep, it looks good.
Thank you. And hello, everybody. So I'm pleased to do this presentation today. So I'll start right away with that question. Why should we identify vaccine candidates in Clostridium perfringens, and how easy or difficult can it be? Well, over the past few years, researchers have looked into necrotic enteritis a bit more and try to better understand the disease pathogenesis and try to identify additional virulence factors. So they took a closer look at the role of spores. They also documented the biofilm ability biofilm formation ability of the bacterium. They also identified adhesion factors or contributors, such as cell surface anchored proteins. The role of bacteriocins has also been documented, and the perfrin bacteriocin was identified. Different enzymes were also linked to any pathogenesis, with chitinases and a zinc metalloprotease getting more attention. And of course, toxins that are the hallmark of C. perfringens virulence have been studied in more details. And more specifically, attention was dedicated to the alpha, NETB, TPAL, and CPB2 toxins. Very few virulence factors, in my opinion, have been identified so far in C. perfringens, and even fewer of these virulence factors have been investigated for the role as putative antigens. Why should we adopt a non-traditional approach for antigen identification in C. perfringens? Where for all of these, those virulence factors that I presented and that were investigated for the role as potential antigens, all of them have been looked at using a classical conventional vaccinology approach, which means, which requires the cultivation of the pathogen and its dissection into individual components using biochemical, microbiological, and molecular methods. And of course, it requires the individual testing of these components, which can be time consuming. A new approach that was described, I would say 20 years ago, is reverse vaccinology. So reverse vaccinology is an innovative approach in the way that it does not necessarily require the cultivation of the pathogen, but can also involve it. And I will present what we did over the coming slides. It is based on the whole bacterium genome sequencing data, and an in silico approach is prioritized, meaning that we will usually rely on bioinformatic tools to identify, characterize, and localize at the bacterium level the proteins of interest. So this approach allows for the rapid identification and testing of multiple candidate antigens. Few words on the comparative and subtractive form of the reverse vaccinology approach. Comparative because this way of using reverse vaccinology will involve commensal and virulent clones of the bacterium, the bacterial species that is studied. And moving to the subtractive form means that what is unique to the virulent clones will be identified and isolated, so we can specifically work on genetic components or genetic determinants that appear to be unique to the virulent clones and that can potentially play a significant role in the virulence of the pathogen, but also in the immunity that is elicited in exposed animals. So this is what we decided to do, adopting a non-traditional approach in the project we conducted. So we first of all did a strain selection. So we selected C. perfringens strains from our unique collection, and these strains were recovered from different broiler chicken farms that participated into a field study a few years ago. So these strains were recovered from either healthy or any affected flocks, and they were characterized using toxinotyping, PFGE. We also established the type 4 pilus profile for most of these strains, and some of them were also tested in a necrotic enteritis experimental model. So in total, 10 commensal and 6 virulent strains were submitted to whole genome sequencing, and to these genomic data, we applied comparative and subtractive reverse vaccinology. So these strains, as I mentioned, were whole genome sequenced. So we did library preparation. They were sequenced, and the data were assembled and analyzed using the different device or softwares that appear in smaller characters. Once we got the data, the genomic data, we were interested in doing protein localization and predicting the immunogenicity, putative potential immunogenicity of these proteins. So first of all, we did subcellular localization using different softwares. We also looked for the presence of transmembrane regions of signal peptides and also for the presence of alternative secretion pathways. And we predicted the immunogenic score for all of the proteins that were present in the genomes we sequenced using the VacciGene software. For the final selection of the candidate proteins from the genomes we analyzed, different criteria were used, but the three main criteria were the presence of the protein solely in the genome of virulent C. perfringens, the localization, as we were interested in working with proteins that were localized at the surface of the bacteria. And we also took a closer look at the immunogenicity score, as we wanted to work on proteins that we wanted to increase our chances of working with proteins that would elicit an immune response in birds. We also decided to include what we call our negative control, so a low immunogenic protein, a protein that was attributed a low immunogenic score using the VacciGene software, and this protein was named P537. So on the right-hand side, you have a table showing the different candidate proteins that were selected based on these criteria. So P264, P509, P153, P561, and our negative control 537. For these proteins, you have the length in amino acids, you have the immunogenic score. I will come back to this element in the next slide, and you have the cellular localization. If you're interested to know more, learn more on what we did, we published a paper earlier at the end of last year in BMC Genomics, summarizing the whole approach we used to identify these candidate proteins. So all of these proteins were cloned, expressed, and purified, and they were used in immunization assays. So proteins were individually tested on broiler chickens that were placed at 1 day of age and were euthanized at 35 days of age. Birds were vaccinated at 7, 14, and 21 days of age, and we collected blood from these immunized birds at 7, 14, 21, and 35 days of age. So how did we try to connect the in silico and in vivo dimension of the project? As I mentioned, for all of the candidate proteins, we looked at the antigenicity. We did antigenicity prediction. We looked at the immunogenic score that was generated by VacciGene. So you can see that this score was pretty high for some of the candidates and decreased for other candidates, with our negative control that was attributed a very low immunogenic score. we wanted to do antigenicity validation using the in vivo assay that I just described. And from the serum, the blood samples and serum that were collected from the immunized birds, we used ELISA to document the antibody response, the IgY antibody response in these birds. So you have the IgY titers for all of the different candidate proteins. What you can see is that all of the proteins were able to elicit an immune response in vaccinated birds. But another interesting observation, and here just above in the red square, you have the different titers. And this value corresponds to the highest titer that was observed in a specific group corresponding to a specific protein. So you have the different higher titers corresponding to each of the proteins. But as you can see, it is difficult to link the predicted immunogenic score to the response that is observed in birds, as for example, our negative control that was attributed a very low antigen immunogenic score generated or elicited an immune response in birds that was comparable to what was generated for the other candidates. So in conclusion, a comparative and subtractive reverse vaccinology approach applied to C. perfringens allows for the identification of multiple putative antigens. Results generated in silico do not seem to correlate with the immune response observed in birds during the in vivo assays. And all the putative antigens identified in this study should be evaluated in a necrotic enteritis experimental model. Well, thank you for your attention, and I'd like to thank our industrial partner in this project, DCL Nutrition et Santé Animale, the founding organisms who supported the project, so MAPAC and MITEX, and the students on the project, Ilem Enyai, Sarah Edarpana, and the members of the Research Chair in Meat Safety Lab. Thank you.
Great, thank you very much for your presentation. That was excellent. We do have a question here regarding the type of ELISA that you use to test your antigenicity prediction. Can you talk about that?
Yes, we use indirect ELISA.
Indirect ELISA, okay. And so now that you've determined that that might not be the best approach, you've suggested that using, you know, all of those types in an experimental model. Is that the next approach that you think will give you better answers, perhaps?
Well, actually, I did not present these results, but this is what we did.
Oh,
so
you did.
Initially, we were planning to, using the reverse vaccinology approach, identify potential candidates and then see if they were able to generate an immune response. And based on this response and the putative score that was attributed by Vaccigene, make a final selection, maybe of one or two or maybe three candidates. But as all of these proteins were able to show interesting results in the immunization assays, we decided to include all of them in the challenge assays. So this is what we did, and we have a paper in preparation regarding these results.
So stay tuned. So you were hoping that it would sort of lighten the load or the number of treatments that you had, but in fact you had to include all of them. So
yes,
very interesting.
So make sure you refine your selection when you use this approach, just making sure that you do not include 10 or 12 or 15 proteins. That was our initial goal. And of course, I listened to the presentation yesterday, presenting the challenges and solutions related to expressing recombinant proteins and purifying them. So of course we experienced these challenges as well. So it kind of decrease or refine our selection a bit more. So we ended up with these candidates that were ultimately all tested in the immunization assays and in the protection assays.
Perfect. Well, with that, I'm going to thank you for your presentation. I don't see any more questions, and we'll continue on with our program. Thank you very much.
Thank you.